Thank you, Natasha, for inviting me. For those of you that know our company, there's a small change to the logo. So now Illumina has acquired our company about 3 weeks ago. So we're part of Illumina. I'm from the dark side. I'm an engineer. So, but we do good things too. So I'll show some of the good things we have been trying to do. So most of newborn screening is typically currently done mainly in this setting, which is in established newborn screening programs. We have multiple samples, mostly testing done on a single function platform, immunoanalyzers or thermocyclers or plate readers. So that's where most of the action is happening, multiple samples, single type of test. But there's a lot of, I don't know, several tens of millions of babies that are not getting tested that live in this side of in this quadrant, which is in the emerging programs where you would need a multifunctional platform and typically a single sample, one sample, many tests versus many samples, single test. And on the way to getting there, there's a hospital lab NBS program where it's more like point of care tests for conditions such as hyperbilirubinemia, where you can't really put it on a blood spot, or galactosemia, MCAD. There are some conditions where immediate results would make a big difference, even in established newborn screening programs. So I'll talk a little bit about this activity and some about this. So needs, newborn screening needs-wise, it's the same: fast time to results, low sample volume, ease of use, portability or small. These are all desired characteristics for any device, actually. But what drives this need is slightly different between, say, a hospital lab setting versus an emerging distributed setting. For example, fast time to results. In a hospital lab setting, it will be really driven by clinically actionable results, such as galactosemia, MCAD, and hyperbilirubinemia. Whereas in emerging settings, it's not only that. In addition, you don't want to lose patients to follow. So typically, the address is not clear, or there are many reasons where the babies are, the moms after delivery are pushed out of the hospital quickly. So you don't want to lose the baby. So that's why you might need really quick results. So similarly, if you look at all these, each of these needs, it's driven by some subtle differences, but the end result or the requirement that drives the product is still the same. You want it to be really small. It should be usable by minimally trained. It should be potentially usable by minimally trained workers in emerging settings. Maximize the sample utility, take a little sample and do a whole bunch of tests. So, and I should say one of the primary limitations is technology. There's no, there are not that many technologies that can answer all these needs simultaneously. So I'll present a little bit about the technology that I had been working on and my company has been working on for a while now. We call it digital microfluidics. The idea is you have hydrophobic surfaces and you can programmably turn the surface hydrophilic. So what you're seeing here in this video is a 300 nanoliter droplet that's dispensed from a couple microliter size droplet. So you can dispense, transport, mix, merge, split, and dispose. So with these operations, pretty much you can build very, very complex liquid handling protocols on a chip. So essentially what we have is an electronic pump. So our instrument doesn't have any moving parts. It's just an electronic pump. By just turning electrodes on and off, you manipulate these droplets. So just wanted to show this video, which shows how we have been able to pull all these operations together to do something. In this case, it's not as complicated as some of the other things that I'll show, but just this is really taking 12 dried blood spot extracts, setting up an array of 12 by 5, that's 60 droplets 60 reactions, 5 LSDs and 12 on 12 samples. It's just an illustration. We set up a whole lot more than this, but this makes for a nice video. 60 droplets, and then each of these 60 droplets is mixed with a stop buffer droplet that stops the reaction. Once the stopping is done, they all line up and go towards a detector and give off fluorescence or color or luminescence, whatever the signal is. So in terms of product platforms, we can actually, I usually like to say this. So we started out with this one actually. We made handheld, and it was just not credible. This was several years ago. Nobody would believe that all this could be done in the handheld. So all we did really, in reality, was we just took that and put it in this box. And now it looks like a real instrument. It feels like a real instrument. So there was acceptance. Okay, yeah, now this must be something. This must be doing something useful. So it doesn't look like a toy. So that's where we went, because that's where the need was and that's where the market acceptance was. But essentially, both of these have exactly the same capabilities. So in terms of the scalability, if a technology can scale between single sample versus multiple samples, you can see that it can scale between distributed settings and into a central lab. So we can take it in either direction. It's only a customized chip that will be different, and the instrument, the casing will be different. Otherwise, the heart and brain of the instrument is the same. And there are some other key differences. In case of a distributed setting platform, you would need to have reagent storage on the cartridge. Whereas in case of a central lab setting, users can load the reagents. And that's a big technical effort to get it all packaged and keep the costs low. Again, in the central lab, or this box is not really that big. The box on the right-hand side, it's about this big. So it can be put on a cart and it's still portable to a certain extent. So in the hospital lab, that's something that can still be used. And right now we are doing a clinical evaluation of this platform at Stanford. It's actually sitting by the NICU, not in a lab. So it's reasonably portable. So the capability has to exist where you can use both dried blood spots and whole blood. So that exists on this platform. And on this one, in a more portable platform, you want it to take directly whole blood and just process it right away. When we started out in newborn screening a few years ago, we were very naive. We didn't know anything about newborn screening. As I said, I'm an engineer, so I had no idea about this whole universe. So we were wondering, why not just go to whole blood and just do it at point of care and give the results out right away? Yeah. So we came up with this platform. So this is 4 instruments tied together to get the throughputs that you would require in a newborn screening centralized lab. We have designed a cartridge that's the size of a microtiter plate. It takes in 48 samples and does 5 different LSDs on each of those 48 samples in about 3 hours or so. We came to realize 8 hours is a good number. So if you can fit in 2 rounds of 2 runs, then you get more runs per instrument. We started making, with some help from CDC and our own efforts, we started making our own QC spots, and now we package the reagents also. It's really, the platform is an electronic platform. It's really plug and play. Just plug it into the wall and it's ready to go. There's no, because this is a microfluidic platform, there's no fluidical HVAC or any plumbing that needs to be done. It's all, plumbing is built in. So typically we use about 100 nanoliters. So we take a dried blood spot punch, put it in a 100 microliter extract volume, and we take only 100 nanoliters per assay. So for these 5 assays, that's half a microliter, and we still have 99.5 microliters extract left behind from a single punch. So this just gives you an idea of the complexity of all the operations that happens on the cartridge. So we set up initially calibration, and then once the calibration is set up, there's a fluorimeter here, so it gives us the signal there. And then this is obviously a little bit speeded up, the whole 3.5-hour protocol in 1 minute. So it sets up 44 droplets times 1, 44 times 2, 44 times 3. So it sets up an array of 44 by 5 droplets, and then takes each of these 44 by 5, runs it by a fluorimeter on the way, mixing with a stop buffer. So it's essentially a robotic liquid handling robot built into this cartridge. Essentially, that's what it does. So Missouri, the state of Missouri, has been doing a pilot. They started a pilot for LSD screening, started in mid-January. It's ongoing right now. They're screening for Pompe, Fabry, Gaucher, and Hurler on the 48-sample cartridge. So far, they have screened 53,000 babies, and I think between Mayo is also doing a comparison study. Between Mayo and Missouri, we've recently crossed the 100,000 baby mark. Nowhere close to the tens of millions that PE has done, but it's a big milestone for a small company. In these 53,000 babies, what was quite surprising is 6 babies have been confirmed for Pompe, 14 for Fabry, and 2 for Hurler. This is confirmatory diagnostic results. It's significantly higher than what's expected from literature. So now shifting into the hospital lab-based newborn screening, which is more like point-of-care newborn screening. A typical workflow would be something like this. This is how we are doing actually in Stanford right now, where at the time of collecting the dried blood spot filter papers, the nurse collects a capillary tube, blood in a capillary tube, and then ships it off to the instrument, which is close to the NICU. So that goes off to the instrument, it gets loaded into the cartridge, and then the cartridge is programmed to do a whole bunch of different types of assays. So we are looking at total serum bilirubin, G6PD deficiency, and congenital hypothyroidism, conditions that are directly related to hyperbilirubinemia and conditions that lead to hyperbilirubinemia. And we are developing a few more, adding a few more tests to see which ones should be a subset. So we are looking at albumin, carboxyhemoglobin, and a few other markers along with this. So far, Stanford has screened about close to 150 newborn whole blood samples, and the comparison looks, this is obviously funded by NICHD. The cartridge has been designed to expand to other assays also, galactosemia, CAH, whatever makes sense in a hospital lab setting. So this is a very complex protocol. So I won't even attempt trying to explain this. So we put in whole blood samples into these reservoirs, and whole blood gets separated into plasma and serum on the cartridge. And we take serum for certain assays, plasma for certain assays, and whole blood for certain assays. So we get 3 types of samples from one input. And then we do an absorbance-based dye-binding assay for bilirubin, and enzymatic fluorescence assay for G6PD, and immunoassay for TSH. All this is done on this single cartridge. And you can see as you go, there's combinations of working reagents, and the sample at 9:1, 18:1, 27, different ratios. It's all programmed into the cartridge. We are turning biochemists into programmers at the company. So they program their protocols into this on the cartridge. So there's a lot of action going on. So while TSH is being detected, G6PD assay is being set up, there's a lot of parallel operations that's going on. So the results look fairly good. So G6PD, we are able to show clear separation between normals and affecteds. TSH is a work in progress. Bilirubin looks very good. Whole blood reference method versus our method. It's the same instrument. It's just a different cartridge programmed slightly differently. Similarly, we are looking at, we are funded by NIDCD to look at CMV from newborn saliva. And this is a molecular assay. So we take a saliva sample from a newborn and load it onto the cartridge. And the cartridge extracts DNA, prepares DNA, purifies it, amplifies it, and then gives a real-time PCR result. So this is 15, maybe 10. Yeah, 10 CMV-affected baby saliva samples, and this is 5 normal newborns, which doesn't show any PCR characteristic curves. The key thing here is to reduce false negatives for newborn hearing screening. This could be useful. Again, the instrument is the same. It has a slightly different detector in there. It's the same as the LSD or as the hyperbilirubinemia instrument. The cartridge is a single sample cartridge, so this is something that gets us towards the hospital lab and eventually into the fully distributed settings. So there are lots of issues to ponder. I just wanted to throw a few of these out there. So in hospital lab programs, I think the issue to consider is how best can we leverage the point of care screening experience from pulse ox, hearing screening, and how can we add biochemical screening tests also. There are lots of complexities in doing that. These are not as non-invasive as the other two tests are, but how best can we use that experience? In emerging programs, there's a lot more questions. So would the screening test be the only test before any clinical action is taken? Bilirubin, for example, would there be a confirmatory bilirubin test, or would this be the only test? So in that case, are we really getting into the realm of diagnostics? Is that still screening? I know in some countries, private pay, it's offered as the birthing package, where you get newborn screening also. So should it be part of that, or would it be part of public health program? So, and who would do the follow-up in the case of this private pay? Test panels will be most likely regional, and what would be the regulatory path? We can do all this technically. That's not I don't see that as the issue. It's really answering these kinds of questions. So we can customize a panel. It's the same cartridge. We just load different reagents and we customize it. But what would be the regulatory pathway? So for each customized panel, do we have to go back and get approvals? What would that look like? Who would do the PT and quality control? This will be similar to point-of-care diagnostics. So would CAP or JCAHO-like organizations do it, or would it still be CDC? There's a lot of questions. So just wrapping back up to the title of this presentation, this is my last slide. So is it driver or passenger technology? 10 years ago, when I was a fresh graduate student out of Duke starting a company, I would have said that, yeah, technology, yeah, that's the way to go. Technology is what we need to. But I think having gone through the development of technology and the full circle of building a company, I think this is, that's the driver for sure, and this is a passenger. That's how it should be. That's my conclusion. And obviously, lots of contributors. Any questions?